part is neuroectodermal cell. Neuroectodermal cell is the portion of the ectoderm of the early embryo that gives rise to the central and peripheral nervous system, which is glial cell.
Neuroectodermal cell consists of glial cells, neurons, endocrine cells, and melanocytes. Glial cells. Glial cells is the supporting cells of the nervous system. It provides support and protection for neurons. Glial cells consist of four main functions, which is to surround neurons and hold them in place. Second, to supply nutrient and oxygen to neuron. Third, to insulate one neuron from another. Lastly, to destroy and remove the carcasses of dead neurons. Glial cells consist of Schwann cells, microglia, astrocytes, and oligodendrocytes. Schwann cells is a supporting cells of the peripheral nervous system. Schwann cells direct nerve axon. It erodes is the single segment of an axon of myelin sheaths. Schwann cells also clean up PNS debris or peripheral nervous system debris and guide the regrowth of peripheral nervous system axon. Next is astrocytes. Astrocytes provide physical supports to neurons. It will clean up the debris also. Besides, it will provide chemical for proper functioning and control chemical composition of fluid surrounding neurons. Besides, it also is a nourishment to neurons. Next is microglia. Microglia is the smallest glial cells that protect the brain from invading microorganisms. It also reacts like a macrophages. The last one is oligodendrocytes. Oligodendrocytes is to provide support to axon. It also produces myelin sheath. Ectodermal cells is neuron. Neuron is the nerve cells. It not survive on the untreated glasses or plastic. It will demonstrate outgrowth on collagens and poly-D-lysine.
Next type of neuroectodermal cell is melanocyte. Melanocyte is dendritic glial cell of the epidermis that synthesize tyrosine. Part of neuroectodermal cell is melanocyte. What is meant by melanocyte? Melanocyte is the dendritic glial cell of the epidermis that synthesize tyrosinase. Besides that, it has been utilized to obtain preferential melanocyte attachment and growth in a hormone-supplemented medium. The supporting good melanocyte for proliferation, which in the high substrate defensivity of cultured keratinocytes. Besides that, absence of chemotherapeutic agent, and then in minimal supplementation of serum, and the last is in tumor promoters, which is probol ester. This part is endocrine cell. Endocrine cell is the body cell secrete substance which influence another cell. Endocrine consists of two parts, which is endo and crine. Endo means secretory products stay inside body, and crine, which is means secretion. What is the characteristic of endocrine? Characteristic of endocrine consists of specialized for secretion, and then their secretory products are secreted into interstitial fluid, but endocrine secretion does not involve that. It also required close proximity to blood vessel, and lastly, The characteristic of endocrine cell is commonly arranged into cord and small clumps.
Hi, Assalamualaikum. And right now I will proceed to the gonad cells. Okay, the gonad cells consist of ovum and sperm. So I will start with an ovum. So there are many cells that can be used as ovarian cells to culture, and one of it is ovarian granulosa cells. And these cells function very well in a primary culture. But when you result culturing it, which means you put it into a different type of flask, okay, the function that was expressed in the primary culture will be not present or lost. Okay, so N of the granulosa, ovarian granulosa cells, and a cell line can be started from a mouse. Not a mouse, but actually a hamster. It is called a Chinese hamster ovary, or CHOK1. And this Chinese hamster ovary cells is cultured for many years, but the lineage of the cells is still unknown. So if you are trying to look it for the lineage of the cells here and there into internet or anywhere, you will not find it. And these cells at some stages, okay, has an epithelioid kind of characteristics, but it can have a fibroblast-like modifications when you are culturing it in a dibutyl cyclic AMP. And other type of ovarian cell line, which is called the human surface epithelial cells. This human surface epithelial cells is isolated, okay, and is immortalized by the HTERT. What does HTERT do? It retaining the function of P53 and also the PRB. As you all know that when HTERT present, the human surface epithelial cell line will escape from the senescence. And some of the human surface epithelial cell can be maturing into an early oocyte. So end of the ovum, we proceed to sperm. Okay, sperm or the testis, human testis, there are some cells that are isolated from the testis, which is the spermatogonial, setoli, as well as the spermatocyte. And these three cells is used as investigation of a hormonal growth control in spermiogenesis. And then these three types of cells is also used to study the cell interaction. Okay, and there are also other cells that can be used, which is the TM4, that is derived from the mouse of a Leydig cells, the mouse Leydig cells, and TM3 is called the mouse Sertoli cells. And then the last cells that I will discuss on is the pluripotent stem cells. This pluripotent stem cell is used as a model for investigation on the growth control, the growth factor control of germ cells maintenance, as well as the spermiogenesis. So last part is I will talk about the culture medium. These cells can be used in a basal culture medium, plus with the DMEM, and then it is also cultured in carbon dioxide incubator 5% with 100% of humidified air, and the medium must be changed twice a week. Okay, this is my references, thanks to the Freshney book and as well as the Oxford Journal. By the way, you guys, this is the members' names, and then any illustrations that meant in this video is not entirely correct to the original. No life is missing, no death is occurring making these videos. And I absolutely thank you to all my lecturers, to all our lecturers, to our instructors. Sorry if we have anything wrong. We love you guys. Bye from Group 3A. See you.